Dear students, we are going to see about quality control and microbiology, that are the standard procedures and standard methods that have to be followed for the quality control in microbiology laboratory. Before that, we can see about the what is microbial quality control, which is the scientific discipline that is dedicated especially to ensure that the products that are from either industries or commercial products that are free from the harmful contamination. So this quality control, it acts as the final safety checkpoints before the product it reaches for consumers purposes. We can use this microbial quality control to detect the microbial contamination, and we can enumerate the microorganisms, and we can identify the specific pathogen is present in the product or not, and we can verify the sterility of the product, whether the product is sterile, that is free from the microorganisms, and we can monitor or manufacturing the environmental conditions that are available in the industries, and evaluation of preservative systems and compliance with the regulatory and quality standards. So these are all the basic introductory concepts about the microbial quality control. So before that, there are several methods are available to ensure that the quality is maintained in the industries and environment. And the main standard method is the pour plate method. The purpose of this method is enumerate the obligate anaerobes and heat sensitive microorganisms. So this is the basic purpose of this pour plate method. As you may know that this we can directly pour the media into the plate, and then we can before the addition of this media, we can first either we can add the samples directly to the empty sterile plate. So this is the pour plate method. And spread plate method, using an inoculation loop, we can spread the sample throughout the media which is already sterilized and poured in the Petri plate. And so this is the purpose and principle of this spread plate method, to enumerate the obligate anaerobes, which are heat sensitive microorganisms. Here the sample is spread over the surface of the pre-solidified agar, which is also sterilized agar, which avoids the heat shock of the molten agar that is used in pour plates, and ensures the all colonies grows on the surface, making morphology easy to distinguish. So we can easily identify the single isolated colonies using this spread plate method. And then next method is membrane filtration, which is always called as an bioburden, and this aims to enumerate the microbes in samples with the low bioburden or large volumes. If we have a large volume of samples, means we can either go for this microbial membrane filtration. And this method, it uses the membranes and it filters the microorganisms by proceeding a large sample volume through a 0.45 micrometer filter. So we are going to use the membrane filter to filter the samples through this membranes. And then MPN test. MPN test is used, that is most portable probable number test, which is used to estimate the density for the samples. So density of the microorganisms can be measured using this MPN test, which is that are insoluble turbal number. So either Enterobacter fecalis can be isolated using this most portable number test. Thank you, students.